So thank you for the invitation. It's an honor to be here and meet you. I have never been to Georgia, but I would love to go sometime. Um, Imagine, is it humid and warm? So mushrooms like, yeah.
Yeah, we went on a hike today and we found at least, I don't know, 12 different mushrooms that were.
Oh, wow. Amazing. So before I get started, I probably have way too much material. I like to see, I mean, not all mycology associations or clubs have a very applied focus. It sounds though like your group does. I mean, with a lot of focus on like growing mushrooms and some of these other applications, which is great. But I thought I would touch base quickly because there's so much I could present on, but I want to make sure it's what y'all are interested in. Can people kind of, I guess we could do the hand thing again.
We need to switch from.
Wait, we'd have to switch to the
oh, got it. Okay.
Why don't you just share everything you want us to know about it, and we'll be super happy with it.
Okay, sounds good. I think so. I was going to focus today on some of the applications for fungi that led, Cornelia Sam and I to meet in the first place. So some of the more environmental work with fungi to address waste and to clean soil and water. So I've also worked a lot on food and medicine projects with fungi. So if that's something you're really interested in, then just, please ask and we can go in any direction. Or if you're really interested in water more than soil, just let me know. We could go in that direction too. But so I just wanted to talk today and share a bit of my journey in mycology with a focus on these applications for fungi in soil and environmental health. And so it kind of starts actually a really long time ago. I mean, when I was a little girl, I was given a little kitchen set, which I immediately put in my closet and turned into a lab to solve world problems. I put a sign on the door. My parents thought it was hilarious. It was something like, do not enter, like experiments in progress. So I've really sort of always been very curious about, what we can do to solve some of these environmental problems that we're facing. So I wanted to share, this is actually where I'm from. I mean, I didn't grow up in Lake Erie, but I grew up right alongside Lake Erie. And so you may be familiar. I mean, Lake Erie is cited in the Lorax. I think the line is, I hear things are pretty bad up there in Lake Erie. So what you're seeing is a toxic algae bloom in the lake. From agricultural runoff. There's also nuclear power plants right on the lake. Second largest plastic gyre outside of Texas, I think, or the big one you always hear about is in Lake Erie. Anyways, there's just, it's a place of a lot of industrial agriculture and other types of industry. The big three auto factories are all there. And so I, really just sharing that for me, I really grew up seeing the problems. I was very, very familiar since I was a child with, the problems that we're facing environmentally and how those impact our health, right? So, and we live in really polluted places and then, our health is impacted. So we are part of Cancer Alley in this region as well. At the same time, probably my first exposure to the power of fungi to help with this came when I was a teenager. And so I would go on these huge walks, like 50-mile walks, all from the agricultural county, from the shores of Lake Erie, where I lived. And I would walk all the way to the nearest city. And along the way, I saw abandoned factories, literally I saw shaggy mane mushrooms shown here. You may have heard that shaggy manes can eat asphalt and oil, and they have the hydraulic pressure as they emerge and fruit to actually push up through asphalt, as is shown here. So I actually saw this when I was a teenager. There was an abandoned or like decommissioned, I guess, oil refinery near where I lived, and I literally saw a ring, like almost like a fairy ring of shaggy mane mushrooms around one of their oil tanks that was left behind, pushing up through asphalt. So it wouldn't be till later that I kind of put some of these pieces together, but I'll mention as well that it wasn't just mushrooms that I saw, on these walks coming into contaminated areas, polluted areas, and helping to, recover, regenerate the ecosystems. I saw plants too. You always see plants coming up through cracks in the pavement and so on. So I think I was instilled with a really strong sense of not only what the problems are, but also how nature heals itself, and how fungi, plants, microbes all come in and together work to regenerate ecosystems and soil and water quality and health. So now I have a mentor, Nancy Clem in Chicago, says life replenishes itself, soil cleans itself, bodies heal themselves. And I really I like to start here because it's sort of the foundation or the philosophy, I guess, of my work with fungi. Whether it's working with fungi, consuming them as medicines, where one of the great things about fungi is they don't have the same, it's not like taking a it's not like taking a pill, like a pharmaceutical pill. A lot of medicinal mushrooms, they tend to not have an action other than reminding our bodies of how to function properly, right? Our bodies know how to heal themselves, and fungi as medicines can often remind our bodies of how to get back into regulation. And that occurs as well in the environment, with soil. Even the most contaminated, barren looking sites have life there, and we might not be able to see it yet, but life replenishes itself and cleans itself. So that's sort of the foundation of and my philosophy for how I do this work. So I had been doing actually food and agriculture work for many years, and finally that's how I got in. So finally the pieces all start coming together. Oh, thanks. And I was helping elementary schools and high schools and other community centers start farms and start growing food, and we would test the soil and find out that it was contaminated with lead and other hazardous metals. So a lot of projects stopped at that point because people didn't know what to do to address these metals in the soil and make it safe again and make it clean again to grow food in. But for me, that was really the start of all these different things coming together, and what really inspired me to start D.I.Y fungi. I knew that fungi played important roles in soil and that they could help, or I had heard that they might be able to help with remediating or cleaning up soil. So I began to try to learn as much as I could about fungi. I knew that to work with them for environmental cleanup, one needed to understand their life cycles and be able to cultivate large amounts of fungal mycelium and things like that. So I started growing a lot of mycelium and I started trying everything I had heard of. I was like, okay, can they degrade oil? check. Okay, can they, you know, I just tried everything I had heard of. And people started asking me like, wait, what are you, that looks cool. Can I have some spawn? Or like, can I have some, yeah, what are you doing? I want to learn more about that. And so I didn't intend to start DIY fungi. It just happened because people kept asking me for mushroom cultures and for education around, you know, how to grow fungi for food, medicine, and to work with them in these, you know, for environmental cleanup or to deal with waste or things like that. So that's how DIY fungi started. And so I did not have, you know, I would say that mycology turned me into a scientist. I once again found myself grown up, an adult with a lab in my closet for solving world problems like when I was a girl, a little child. And so yeah, I worked on a number of projects over the years. That was DIY Fungi was founded in 2012, so it's been going on 10 years, lots of projects now, and ultimately, you know, the path led me towards this PhD program in environmental toxicology, where I study fungi as well. So I thought today that we would take a little journey through some of the projects I've worked on, and you know what I learned, and what I'm learning today in my current research in Los Angeles, trying to really apply some of these fungi to clean up large and heavily contaminated sites and do some field testing of some of these methods that seem pretty promising on a smaller scale. So, oh yeah, I guess I'm just going to mention this here too. I said that when I started doing stuff with fungi, especially, or I guess I was doing food work, found out that a lot of soils are contaminated with lead and other metals. I was like, what can we do? We got to figure out how to deal with this. But one of the barriers to dealing with that issue and to remediating and making sure that the sites are cleaned up is the expensive nature of soil testing and different types of testing. So I founded the Healing City Soils Program. It's a partnership with several universities and a compost education center. And we offer free, it's still running to this day. We offer free soil metal testing for people growing food. And I just wanted to mention this here because throughout this presentation, I'm going to be really sharing different examples of projects that were done in a community science way, again, with no funds really, or materials or special laboratories or anything like that, just total community kind of DIY. And so I'll also be sharing examples of projects that were done in a proper laboratory, so to speak, in an academic university research context. And so I just like to bring in this example as a way that if you're working on projects, whether it is regarding, mushrooms as food, medicine, or for remediation, you might want to test them to see, whether it's the nutrient content, the medicinal value, the safety, things like that. And testing is really expensive and can be difficult, but I just like to share this to say that there are ways through building partnerships with universities that you can often access testing and analysis of your mycological research projects. And there's a huge benefit to that on both sides, right? It can really, so I just wanted to share this for those of you who are pursuing your own experiments with fungi. Reach out to universities and see if they would run some samples for you for whatever you need. So I know that this group, I did not need to tell this group that you can, divert non-toxic waste from the landfill as substrates to grow mushrooms for food. It sounds like you are already doing that, and I'm so excited to hear that. So I'm not going to talk about that too much, but I'm happy to answer any questions. Worked on some projects with this, trying to, again, set up partnerships with like coffee shops and larger institutions that do a lot of purchasing of mushrooms. Worked on projects around this kind of stuff all over the world, and happy to share more about it. But really going to focus today on the piece around applying mycology to reduce the toxicity and accelerate the decomposition of polluting waste, and to remediate and restore contaminated sites. So I like to call this one person's trash is a mushroom's treasure. And, some people say that waste is just a misplaced resource, and I fully agree with that. Globally and in the US, there's widespread food insecurity. People do not have access to enough healthy food, and protein is one of the major lacks. We have widespread environmental pollution in the soil, the air, and the water, and our landfills are filling up. So trash is being shipped all over or incinerated. And I'm sure I've done this everywhere I move. This is very nerdy, but I pretty immediately do 3 things. One is I research what's in the waste stream. It's the same everywhere, more or less, give or take a bit. We have almost 40% organic. That's typically just food waste, or like yard waste, things like that. It's organic material. We have a lot of paper, and then a good amount of plastics as well. But my proposal is to try to transform those non-toxic, like organic waste, like paper, any sort of organic material, which can be a substrate for fungi, into food. And then to intersect and collect and transform some of these other toxic waste materials into soil, into clean soil. And I'll talk about some projects I've worked on that have done that. And just to finish, the other 2 things that are also very nerdy that I often do as soon as I move to a new place is check out the Mycological Society and try to connect with them, meet other mushroom lovers. And the third is that I start to go and getting little samples of plant roots and staining them and putting them under a microscope to see if there's mycorrhizal fungi. So I'll talk more about mycorrhizal fungi later. Okay, so on the waste, and this ties into the project Cornelia mentioned. So there's the problem of all the waste that ends up in the landfill that really could have a better use, that could be a resource. But there's a lot of common pollutants that don't even make it to the landfill, like oil, cigarette butts, lubricant oils, failing septic, that's another one. But then there's others like plastics and, different products made of plastics, from diapers to, all sorts of other things, textiles, that just don't break down. They go to the landfill and they take up a lot of space, but then they just sit there for hundreds of years, causing problems for our climate and, leading to pollution and things like that. So, so as I mentioned, diving more into the diversion to grow food. As I said, I'm not going to focus on that, but ask me about it if you want to hear more about the projects I've worked on, trying to get my university to supplement their beef burgers, of which they serve a huge number every day in the cafeterias, with mushrooms. that are grown on the coffee waste produced on campus and projects like that. But really gonna hone in on this remediation of waste piece. So to start on that, just wanted to sort of define some of the things to watch out for in wastes. This is also going to be true for soils. So if you are wanting to, grow food on certain wastes, grow mushrooms is what I mean as food on certain wastes, or you're going to be trying to remediate them in some way with fungi. It's good to understand that the two basic categories of contaminants. So one is metals, I've already mentioned. They are inorganic. They're what you'd see on the elemental or the periodic table, sorry, they're elements. So like lead, arsenic, nickel, cadmium, chromium, mercury, and so on. What that means is that, they're inorganic, so they cannot be broken down. They're not carbon-based. So once lead is in the environment, it just accumulates or it moves around. It doesn't degrade. So you cannot, if the waste material you're working with has metals in them, then you need to be mindful that you're going to need to either bind those so they're not getting up into the fruit bodies that you may consume, or just moving around causing harm, or you're going to need to extract them and then contain those metals. And I'll give some examples of that later too. This is different than organic contaminants. Organic contaminants are carbon-based. That means that it's just dead stuff, basically, usually dead, dead plants that got compressed for millions of years and turned into the petroleum products that we make just about everything out of now, from plastics to fertilizers and diesel and so many other products. So organic contaminants can sometimes be actually transformed or broken down, degraded into less toxic or non-toxic forms. So these are the ones that can actually turn into soil, and fungi can often really help with that. And the reason is, quick question, you could drop it in the chat or just speak it out. What do a lot of fungi eat in nature? What is their favorite food? Like decomposer fungi, I mean.
Cellulose.
Yeah, cellulose. What else? What about what makes trees rigid and hard?
Lignin.
Lignin.
Yeah, okay. So basically they eat plants. They eat dead plants. Decomposer fungi, they love eating dead plants. What they really want is the cellulose and lignin, as folks mentioned, that's in there. And so the reason, and they do that, you probably know, but they do that by just excreting these digestive enzymes and acids into their substrate, into their environment, to break down that cellulose and lignin or lignin, so that they can, get the carbs they need from it. So a lot of these decomposer fungi will actually eat or try to eat organic contaminants, because they're really made of essentially the same thing. They're really made of dead plants. It's just that those dead plants were compressed for a long time. But essentially, and I'm just showing these different, like this is the structure of cellulose and lignin. You can see lignin's super complex. I could get into the chemistry if you want, but just ask if you want that. But I'm just showing like how similar structurally they are to some common organic contaminants. Like this benzoate pyrene is a pretty nasty carcinogenic polycyclic aromatic hydrocarbon. I hope I didn't lose you on these big words, but they basically get put out in the environment from car exhaust. And here we have a pesticide that you probably know, DDT. Anyway, so these decomposer fungi will apply those digestive enzymes to try to eat or certain organic pollutants and try to get food out of it. And that's why when you hear that, like, oyster mushrooms or other decomposers can remediate or clean up organic pollutants, really that's what's happening. They think, hey, I might be able to get some food out of this. I'm going to throw my digestive enzymes out there and try to break it down to get some food. And it turns out that breakdown process is really helpful for us because it can help clean up our environment.
Danielle, someone did ask a good question. Lori asked a question about whether neonicotinoids can be broken down by mushrooms.
Hey, so neonicotinoids that's in a type of, what is it, pesticide? Can you,
Systemic. Yes.
That hurt bees. Yeah, I actually looked into that a while ago, and I believe I did find some studies about soil fungi, common soil fungi degrading neonicotinoids. So I believe I have seen some work on that. Oh, cool.
That's great news, because they're absorbed by the plant material and they stay in the plants even after that.
Right. They're designed to be systemic through the entire plant, and they of course affect pollinators. So
Yeah, I hate them.
Yeah, they're pretty nasty. I'll try to dig up those. I'll try to dig up those studies, and I can email them to Cornelia to send out to the group.
great.
I just actually, while we're paused, I just see some really cool comments. I love it. Thank you, everyone. This is funny. I love it. So Sam asked, how are you getting funding for the heavy metal testing for food growers? So actually, the program, okay, we have gotten some grants to be able to extend the work we're doing, which I'll talk about later, because we're actually doing bioremediation now. But the reason the heavy metal testing is free is because there is an environmental science program at the college that we partnered with, and their students actually have to do, every year the students have to do a final project. So we just, every year we propose this project to their student teams, and it always gets picked. And so basically it's a win-win. Students get employable environmental experience in their community, going out, sampling at people's gardens and farms, and then analyzing the samples. And then we all win, because then it's free. The university has the instrumentation to do the testing, and it's free. So yeah, so luckily the program is free just through that partnership. So that's something you could explore where you live. And yes, I will share the link to the bioremediation fact sheet later. But it's available. Yeah, I'll send the link later. Okay, so now a couple of different approaches you can take here. One is that you can acclimatize a fungal species to be more efficient at metabolizing or eating a given pollutant. This is also true for all of you who just grow mushrooms for food or medicine, right? You know this. You might have access to a less preferred substrate. Maybe the first time you try to grow a species on that less preferred substrate, like, I don't know, like, what was someone, I think Katya was saying earlier, or someone was saying earlier, there was something that didn't grow super well. But you can always, sometimes it just needs a little experience with a less preferred substrate, something that's harder for it to eat, and it'll often figure it out. And so the way if you want to do that for any substrate, that you have access to that maybe the species you want to grow doesn't love it right away. The way to do it is you just slowly increase the amount of that less preferred substrate or even contaminant that you're exposing the fungus to, and it will get faster and more capable of either of metabolizing it or dealing with it. And this is true for metals. There was the same strain of this common soil fungus, trichoderma. Actually, mushroom cultivators know it. We hate trichoderma. It's the green mold that can really contaminate our growing kits and stuff, but it's an amazing remediator. And yeah, so this, and you're seeing these petri dishes, and it was spiked with increasing concentrations of arsenic, the metal, over time, and very quickly it became able to tolerate like 700 times the quantity of arsenic. And then here on the other side, we're seeing I was trying to acclimatize a Pleurotus species to metabolize a particular bicycle lubricant. So you can always take the approach of choosing a known fungus that is known to be a pretty good remediator, and then trying to acclimatize it even to become an even better one. Or you can, this is really, my work has really switched to this approach more and more. You can find fungi that are already adapted, that are growing on toxic waste or contaminated sites. Oops. And then work with those. So I'm showing this pile of electronics waste because you might think, oh, there's nothing growing on there. It's just electronic waste. But actually, there's tons of life on there. There's fungi and bacteria. So this is a study that found a fungal strain from an e-waste recycling facility that is a really strong at sorbing or like binding onto different toxic metals. So a common protocol in remediation efforts with fungi is that you collect the fungi from the local environment or waste that's contaminated. You isolate that strain, and then you basically continue to bulk it up and expand it so you can reapply it on the site. I love talking to mycological societies because there's less background to explain. But so, some of the best fungi typically for dealing with waste are the decomposers, but not just the sort of charismatic Basidiomycota, like oyster mushrooms, but also really unpopular, really common soil-dwelling molds and yeasts from the Ascomycota, including like the trichoderma that I mentioned that we hate as cultivators. It's an amazing remediator. As well as endophytic fungi, the fungi that live inside of just all the tissues of plants. Such as the one you may have heard of, Pestiloptosis microspore, which was isolated from a plant in the Amazon that can eat polyurethane plastic as its only carbon source in the presence and absence of oxygen. So that's pretty cool. That's a fungus that was found inside of a plant. So bringing it all together, there's this model I've been developing over the last 9 years or so. I call it mycodigesters. Lots of inspiration from Trad Cotter, Jay Schindler, and Peter McCoy on this. But here's just some photos of the project Cornelia mentioned, it's not just bicycle lubricants, but machines in general require lubricants to function. They're made out of fossil fuels. They're petroleum products that often have Teflon added in to just help the product But Teflon, as you may know, is part of that family of forever chemicals. The PFAS, they basically wash off and then pollute the environment and all life, basically, and then don't break down. So there's still actually a really recent update. There has been a bacterial strain that identified at UC Riverside, the school I go to, that can break some of the bonds in PFAS chemicals, but no living organism had been found until then that can do this. The bonds in these PFAS chemicals, including Teflon, are the strongest in nature. And so it's been thought that nothing can break them down. But I thought, well, but what about what about some decomposer fungi? Because they're pretty good at breaking stuff down. So yeah, so I basically tried out the system. It worked well. I started really small in 5-gallon buckets and then scaled up to these 55-gallon myco digesters that were left on site at the bike shop. And so basically they would soak the rags. I had Pleurotus species, so oyster mushrooms, within the digesters that would grow to consume not only the rag that the Teflon based lubricant was applied with, but also some of the components of the Teflon and the petroleum parts of the lubricant. So I will say I did start with an acclimatization process. It worked anyways, but without it, but it was a lot faster and more resilient when I did pre-expose, like I'm seeing I just did it in jars. I did pre-expose the species to the contaminant, so it had a chance to yeah, just get used to it, get better at dealing with it. I'm going to talk a little more about this in a minute, but another project I worked on, I was actually invited to work on this project by a municipality that's main industry was tourism related to its pristine beaches. And cigarette butts, you may know, are the most common, contaminant or waste item on beaches, but also just around the world. They're small, but they contain thousands of toxic metals and those PAHs I mentioned before that come, they come when stuff burns, basically. So they actually invited me to work on this project with them to see whether fungi could degrade cigarette butts as part of their efforts to collect them and get them off the beaches. And so Peter McCoy had already shown just in a mason jar that, it could be done. And then Alex Dorr did a little bit of testing of that. So I developed an experiment with different inoculation methods, liquid culture versus grain spawn, different like sterility, either sterilized butts or non. And I basically tested the capacity for these Pleurotus ostreatus fungi to degrade the pHs and the other chemicals in cigarette butts. And so again, on this, I partnered with the chemistry department at a different college, and they were super excited. They got their students involved in doing some of the analysis. And we actually found, so I'm showing here, the controls are shown in blue. So basically the controls mean that there were the cigarette butts treated in the same way, but they just were not inoculated with the fungus. And so basically the levels remained high for the untreated ones. And for most of the PAHs, there's a lot of different PAHs that are hazardous. The treatment, whether it was sterilized or not, with grain, inoculated with grain or with liquid culture, there was a significant reduction. I forget offhand, but it was between a like 70% and in some cases like a 97% reduction in only 2 weeks of the contaminants of concern in these cigarette butts. So, so through some of these experiments, I developed this model for dealing with common toxic waste. And I've played around with a lot of different ways to do it. And I actually found the best way was to grow oyster mycelium on cardboard and flat sheets. Of course, you got to collect and your waste and then soak it. So I would just add water from the hose in a sealed container. Cultivators know your substrate always needs to be moist for the fungi to grow on it. And then basically add that soaked waste into the digester. So here's an example of one of these digesters for cigarette butts. And you can see it's not fully grown, but the mycelium is starting to consume the butts. You want to ensure there's adequate air exchange. I learned that the hard way. The fungus is going to need everything it usually needs to survive, right? So it needs air or air exchange. It needs its substrate to be moist. And yeah, you can either add the contaminated material to the top or you can layer it. Both work, but in general, I found that, and this makes a lot of sense when you think to nature, right, and how nature works. But typically these primary decomposer fungi, like Pleurotus species, are really good at starting a process of decomposition. They have enzymes that can break apart really complex, big chains of chemicals into smaller bits that then now other fungi, bacteria, even worms can finally they can like get in there and go to work on ultimately transforming this toxic waste material into soil. So I did find the model works best when you do a primary decomposition with the primary decomposer fungus, sometimes a secondary decomposer fungus, and then some kind of composting process with worms and bacteria. So this is another example where it's really important to be able to test to make sure that that the toxicity is gone, right? Yeah, so that's where partnerships with universities or grants to do testing can really help. I will say I've picked up the lubricant project again recently because there's a lab at my school that studies PFAS, and they have all the ability to do all the proper testing. So I'm really excited about that. But I will mention too, for folks experimenting just on a, just experimenting on a small scale at home, take care of yourself. Don't expose yourself to toxic stuff. Please, wear masks and gloves, and yeah, keep yourself protected. But the bioassay is a really simple way to, you use life to tell you whether the toxicity is being reduced. So that's what I did with the bike lubricants project. I didn't have funds to test at that time, but I did use a worm bin as a indicator that the toxicity was being reduced. So, when I put the bike rags with the Teflon lubricants, if I put them, directly into a worm bin, the worms would not survive. But once I had the oyster mushroom get through it, then the worms not only survived, but they loved it. They would crawl through and turn the whole thing into soil. So that to me was an example of a bioassay that's using life to indicate that, toxicity must be reduced. This is a current project I'm working on that I'm excited about. There's a textile artist that approached me. She's Chilean, and she's really concerned about the textile waste crisis, where, textiles, including clothing and, carpets and all sorts of stuff, are mostly made of plastics these days, and toxic dyes and all sorts of other stuff. And they're a huge portion of the mass of waste that ends up in landfills, but also a lot of it gets burned, even stuff from secondhand stores. Most of it doesn't sell, and then they incinerate it or literally ship it to countries like Chile, where it gets piled in the desert and just left. So it's a huge problem. And so she approached me on this collaboration to work with fungi, decomposer fungi, to transform this textile waste into soil. And I already knew that that could happen because even, with the bike rags project, I was growing on textiles, and I saw the fungi be able to decolor, meaning sort of deal with the dyes in textiles, and ultimately create soil out of those, even contaminated textiles. So yeah, I'm really excited about this project. We're continuing experimentation currently. So just a couple best practices to close this section. So you build relationships and can collect and contain waste, right? Don't hesitate to reach out to your local municipality, businesses, communities, community centers, and work together with, universities. That's a really good one. Universities and colleges, to develop relationships, to collect waste, to raise awareness of the issue, and to access testing and other resources, and just be able to, really ensure that your projects are having an impact. Secondly, ensure the fungus has what it needs to grow, especially water. It needs nitrogen, some easier food sources, and air exchange. Mimicking nature works best. So like I said, in nature, we don't have monocultures usually. It's usually many different organisms all together, all in their own phase, right? Usually one organism's life creates food and habitat for another organism's life. So it makes sense to mimic that when we're trying to regenerate soil or water, or create these little microcosms where we're dealing with wastes. And then you'll have to, usually we're working with mycelium when we're working on remediation or waste management with fungi. But if mushrooms do form, you'll want to have a plant, you'll want to contain them and dispose of them, right? Especially if there is metals involved, because mushroom fruit bodies often accumulate or hyperaccumulate hazardous metals. Okay, so I'm going to take a little pause here before we go to the next section and look at the questions. So yeah, that's a really cool fact. Oops. Ronald, let me see. Yeah, Ronald says all of the coal in the world is between 360 and 300 million years old. Plants developed lignin about 360 million years ago, and it took fungi 60 million years to develop the ability to process it. That's when coal production stopped. Really neat. Sam says, oh, cool. So you've had Jay, Peter, and Trad all talk. Yep, and Trad has, yeah, and in Trad's book, organic mushroom farming and microremediation, he has a couple methods that he outlines for bioassays with beans and worms, I think, as well. You can bulk up. I definitely use submerged fermentation for bulking up. Absolutely. So so for those who maybe are unfamiliar, that I'll do that in like garbage cans that are clean and don't have holes, and whatever the substrate is, you basically fill the garbage can with water, and submerge whatever your substrate is, put a brick or something on top, put the lid on, and let it sit for a week or two. It smells terrible if it has worked. It is a fermentation process where it's mostly anaerobic, so there isn't much oxygen in that system. So a lot of the competitors to fungi die they need oxygen too. So they get sleepy or tired or weak in the absence of oxygen. And then when you take off the lid and drain it and inoculate your substrate, then the fungus you want to grow has a head start. But I will say I've never, and thank you, I think maybe where this was going was like, do you have to sterilize contaminated substrates or contaminated waste? And the answer is no. So again, so with the only time I test, so with the bike rags, I did not sterilize them. I just soaked them like for 1 hour, just so that they were moist. The water, I continued to reuse that dirty water, and I would use it to water the system. So it did not produce any like contaminated wastewater. And then when I tested, the only experiment I tested was the cigarette butts. And remember, I compared sterilized versus unsterilized, and I actually found no statistically significant difference between them. So they grew, the fungus grew, whether sterilized or not. So you do not need to sterilize in any way through submerged fermentation, kind of pasteurization or sterilization of contaminated substrates. You just do need to soak them, which can produce a wastewater, but I would just recommend pouring that, like using that to water your system so that it doesn't end up being this wastewater that you have to deal with.
Really cool. So I always have thought about submerged fermentation as in these huge stainless steel vats inside of a lab, but you're doing it totally DIY.
Yeah.
That's so cool. That's the first time I've heard somebody say it that way.
Yeah. I wish I had, I do have photos of this stuff. It just doesn't look very cool, or like, it just doesn't look very good. It's like pictures of buckets and barrels and stuff, but yeah,
exciting to us.
Yay. I guess I will make a note though, because I've talked to Peter and just others, and they, sometimes people are like, but that doesn't work for me. How does that work for you? But I think the reason this has worked for me is because I was never sterile. The cultures that I have worked with were always exposed to competition, pathogens, everything. So I was, in other words, I was growing and expanding mycelium, growing mushroom cultures in very non-sterile conditions. And I think that produced fungal strains that were very, street smart or something. Like they could handle themselves in the presence of, in real context, like soil, which is full of other microbes. I think with genetics and epigenetics, there can be a bit of a use it or lose it thing. It's not that it's totally lost, but if they haven't used it in a while, if they haven't used one of their capacities to fend off competitors or or pathogens or predators, then they can be a lot slower to kind of like remember how to do that. And I think a lot of mushroom cultivation occurs in perfect conditions. They've only eaten a perfect blend of nutrients and you know, food their whole lives. It was perfectly sterile. They had no challenges ever. And then if you take that type of culture and put it out in some dirty rags that are contaminated, it is not going to probably do very well. But so if you are, you know, I do, and I, you know, and sort of on that note, and I've said this before in interviews and stuff, honestly, I have always found that this is a great use of your contaminated kits and efforts. Again, trichoderma, the most common contaminant on grow kits, is one of the most documented powerful remediators. So with some of these projects, I used, because I had a spawn business, so you know, we always have a bit of contamination, and I would save those. Usually the culture is fighting it, the fungus is fighting it, but there's some contamination on there. And those ones, that combination or synergy between the trichoderma and the oyster often produced really good results.
That is really awesome. Like what you're saying is, it's kind of like, you know, you can't let a cat that's grown up all its life indoors out on the street. It'll just get run over.
Exactly.
Yeah.
Yeah, it's the same kind of idea here. And I honestly think that's why, you know, sometimes there haven't been too many studies that have done applying fungi in sort of on real world scenarios where contamination exists.
Right.
But sometimes they don't work. And I think part of the reason, I think there's two main reasons that it doesn't work. And it's not because it can't work, but one is because they didn't provide for the basic needs of the fungus, right? So an understanding of fungal, you're all in a good place with that, right? Understand fungal ecology. You got to make sure the fungus has its basic needs. And then the second reason is because they cultured, again, they had sterile cultures in perfect conditions, and then you put them out in the real world and it just wasn't able to compete.
This is so great. This is so great to hear, Danielle, because we have heard a lot of those people who've done the work with these single sterile strains and say, well, it doesn't really break down, whatever. And they haven't gotten the street strains.
Yeah.
This is really great.
So there's an advantage to the kind of DIY low budget.
Great.
Yeah. So just to kind of,
I'd like to ask a question.
Yeah.
Hey, thanks. You're doing great. You mentioned that pile of rags, the fast fashion pile of rags in the Chilean desert. And I'm really taken with that idea that it's just out there. I mean, what a desperate image. But imagine that you were given that project of trying to remediate that or decompose it in some way 100 years from now or 200 years from now. What would the best practice be? I mean, would you just like, you know, grow the mushrooms on the substrate, you know, whatever condition it's in, and then remove them to a landfill, or just let them grow, die, and run this course over and over again? Thanks.
Do you mean like with the actual pile in the desert in Chile?
Yes.
Okay.
Yes.
Yeah. I mean, the thing is that you typically want to you need a pretty good inoculation ratio. In other words, like you need a lot of mycelium if it's a huge pile, however many, you know, however tall, it might be multiple stories tall or something. So you would either have to as you suggest, well, I love this kind of thing. I'm just sort of brainstorming right now, but there's a few things that I would suggest. One is you could, if you had the machinery like backhoes and stuff, you could actually sort of layer. So in the past with bigger projects, you'll inoculate, like you'll get big rolls of burlap and soak it. They do typically try to sterilize that, steam sterilize it, and then you basically inoculate it and roll it back up. So it's, you have these huge flat planes of mycelium, and those are really handy. So then you would have the backhoe or whatever basically layers of this, the textile waste with these layers of the mycelium, so that there could be, so the mycelium could adequately grow through it. If that wasn't feasible, then you could try just top inoculating and then letting it continue to, because as soon as, you know, y'all have seen this, I'm sure, as soon as a fungus, like a decomposer, is out of food, it's gonna fruit, spread spores, find more food, right? So that might work. I think that would take longer. Another option would be, and I've done this with soils that are really compacted, is if you could somehow, and you can do this with cardboard or pipe, but if you had like a perforated pipe, and that you could somehow like drill down in, and then you stuff that with either, you know, you use a liquid mycelium, that's another idea, which could infiltrate, or you basically stuff that so that the mycelium can really kind of get in there. Those are just some ideas. But the project I'm working on, we're trying to intercept it before it goes to the desert. But you're right. I mean, you know, turn it into soil where it is. But yeah, you're right. There still is this issue where there's this just massive pile of plastic basically in the desert in Chile. So,
Can I just say something sort of amusing and weird that came to my mind? It's like trying this on a small scale. You could do it with a bunch of Barbie clothes in a small tube and see if it works. You know,
yes.
it's just, I mean,
no, that's a great design. That's definitely the way to do it. Start at a small scale, see if it works, and then keep scaling up. So I'm just gonna, I see a couple other questions. Love to hear more about the beans. Okay. What disposal for the remaining contaminated? So I mean, ideally, oh, do you mean like if there is any contaminated byproduct of the remediation or the waste? Yeah.
We had Alex and Lyra speak last month, and Alex talked about at least a lot of the cigarette butt remainder that he had still had, you know, contaminants in them.
Yeah. So that's where there's a bunch of options. So one is that I've done a lot is you'll just, again, I just use a garbage can with no holes, and you just continuously anaerobically compost them down. So you just put them all in the bin. The biomass is reduced. So you go from maybe a lot of biomass to a really small amount of sludge. And then you can pay, presumably the reason you're needing to do this is because there's metals, which again, don't degrade.
Right.
So yeah, but you end up with reducing the biomass to a really small amount that you can pay to have disposed of at a proper facility, a hazardous waste facility. Alternatively, I mean, there's some really neat stuff happening with this. You can also incinerate it, and then you end up with ash. And sometimes precious metals will be re-extracted from that ash. Actually, kind of surprisingly, I mean, apparently the main use in modern times for lead now is in batteries. And apparently the majority of lead is already we're no longer, you know, mining it. We're re-extracting it at this point. You can do that also with plants, which can extract them into their, so the reason you'd want to do that versus the containment or incineration is if you're trying to produce soil and you want to be able to use that soil, it's otherwise healthy, but it has these metals. You can work with phyto extractor plants that would suck up the metals. Then you would reduce their biomass and dispose of those metals. But then you'd be left with clean soil.
Cool. Okay, thank you.
That's really great to hear. So I get I'm a pediatrician. I get journals, and I was just reading one today that said that half of US children have lead levels that are above what should, you know, recommended. Half of our children have that high, and our tolerance for how much lead a child should have is set higher than it is for other countries, actually.
Yeah,
So
definitely.
that was really sobering to read that today.
Yeah. So I see a question. Has anyone tried to extract metals from the superaccumulate? Yes. And so actually there's this, I'm forgetting what the company is called. This is beyond my technological capabilities currently, but yeah, there is a company, there's more than one company that grows. So, you know, earlier I showed a picture of electronics waste, and there's fungi that have been isolated from that waste that can, they're just superstars at binding up the metals in electronics waste. There are companies that will use fungi to re-extract gold and other precious metals from electronics waste. Yeah, so there's all sorts of neat stuff happening with that. I actually helped some grade 8 students locally with a project. It was so cool. They had spiked a bunch of grow kits of oyster mushroom with different metals, and then they came to me and I helped them test the fruit bodies to see the extent to which the mushroom fruit body had accumulated the metals. That was really cool. There's still more to figure out about that. I think we generally know that some fungi tend to hyperaccumulate metals in their fruit bodies more than others, but it's still not, I think there's still a little more to figure out about that.
Yes, I remember I was reading about superaccumulation in plants. That was before I actually studied geology. And it was really cool because they were thinking about searching, indicator searching for minerals by using plants, and then how to use them to extract some metals from that. But that was in the 80s.
Yes.
So.
Yeah, and I was gonna talk about that a bit more too, because I work with plants also in my research. I think that somebody had a question they wanted to say out loud. Feel free, Frank. Frank, did you want to unmute yourself and ask your question? Oh.
Oh, I'm not unmuted. So Lori, is Frank talking? Because we can't hear him. Maybe you're having some tech issues.
Okay.
Do you want to go ahead and type it in the chat then?
Sure. And then so maybe in the meantime, yeah, I see Jeanette Hansen's question. I love it. The largest scale bioremediation project using fungi that you know of. I would love to share about my current research. I don't want to be arrogant, but I am trying to do it on a large scale because I think there's a real need for that. I haven't seen some of the famous, amazing sort of mycoremediation studies that have been done were done in real conditions, but on a very small scale, like in 10 yards of contaminated soil and things like that. So I'm really excited to have the opportunity to try to scale that up and see how it works in that sense. So maybe just in the interest of time, I will chug along and talk about some of my current research on large contaminated sites, and then and we can always do some more questions at the end too.
Great. So
Okay.
I did get, Lori, they sent me the question. I think Frank's question is,
Oh, okay.
what can a mushroom club do to cheaply test out mushrooms for toxins or metals from an unknown area? Like, and you know, you
Yeah.
had mentioned teaming with the university, that there are students there that have to do projects, and you're just handing them one.
Yeah. Also, I don't know, I'm sure you have an agricultural extension office somewhere nearby. Every
around.
town. So they often have the same analytical equipment that I have in the lab right now, that can test for metals. And so you can, it usually does soil, but I've used it for mushrooms if they're dried and powdered. So you could always ask Ag Extension, you know, mushrooms are a crop that you're eating. So it's, you know, you could make that case that you need to see if they're safe.
Nice.
If that doesn't work out, then yeah, definitely find a lab that does stuff with metals and in a university, and hopefully they'd be willing to run some samples for you. If not for free, maybe they'd want to collaborate, or it would be, sometimes it's pretty cheap, especially through Ag Extension. It's like $10 or something, but so worth starting there.
Cool. Thank you.
One quick question. What is the best way to use mushrooms to deal with compacted subsoil? Because
yeah,
we don't have much soil here. It's being depleted. And when you start digging to make a new yard, it's clay, but it's so compacted.
yeah.
And even oxidized. It's horrible.
Yeah, what I would do, well, you're growing mushrooms, right? Like,
I started dealing with the soil problem, and this is what led me to the mushrooms. So I read
that's
the
happened for me
running,
too. Yeah.
and literally ordered a free truckload of mulch and just spread the mulch on top of the soil. And
Perfect.
mushrooms started coming. Right now it's stinkhorns, but who cares?
Hey, good. Okay, well, honestly, yeah, applying wood chips is a great option. If you want to, I think some of you were talking about what to do with like spent mushroom kits. I mean, that's a great use for it. You can either use them to make hot compost. That would be the carbon part of the hot compost, or you can just literally layer them in with your wood chips. Another good one is King Stropharia. Are you aware of this one?
I ordered this one in January.
Okay.
I sprayed it with fresh wood chips. I haven't seen a single mushroom yet.
Oh, that's strange. Did the wood chips stay moist?
Well, not in the summer, obviously.
Okay, but they should have had enough time. Can you see mycelium?
I see mycelium when I dig into there, but I still don't see any mushroom.
Okay. Well, it's good there's mycelium. Yeah, so that's a really good option. Yeah, I mean, if it's very compacted and very clay, it will take a long time, like many, many years, for natural processes, worms, fungi to.
There are no worms.
And there are no worms, yeah.
With some plants and radishes, but I mean, if there is any way to speed it up with different
Katja, can we let Danielle finish, and then we can come back to you at the end? I will say that right now is the best time to collect leaves. Like, leaves is a really great thing to add to your compost pile, and you can get tons and tons of them all over the city. You know, leaves are free and they're a great source of organic matter. So anyway, but I'd like to let Danielle finish what she wants to finish first.
Okay, thanks. So I'm gonna just sort of go a little quick. Okay, yeah. So let's see. So, okay, in terms of actual land remediation, the most successful approaches definitely mimic nature, of course, right? And it's not just fungi, but fungi often start a process of regeneration that can help and a whole ecosystem recover. So again, here there's two approaches, right? One is you go to the contaminated site and see the fungi, plants that are already growing there, and then try to amplify them. Same as the waste, right? There's probably, it might look barren, but I'm sure if you got some samples and looked under a microscope, you would see tons of life. Or you can introduce known remediators that the best practice here is not to introduce a known remediator from a different climate, from a totally different context, like that grows on wood, and then you put it in soil or something. But it does make sense to introduce known remediators from similar contexts. So for example, maybe you've gone out, you've seen the same plant fungus combination growing on multiple contaminated sites in your area. That one might make sense to reintroduce. And you might need to do that because while there is life everywhere, and intensively managed lands like industrial agriculture contexts, degraded soils like mine sites and brownfields, you might want to either amplify and support the fungi that are already there by adding organic matter, or reintroduce some of these fungi. So I'm just going to really quickly mention a project I've been working on out in Southern California, where I went out to contaminated sites all over. It's an arid area here, and I basically looked at the plants that were, oh, this got weird, that were showing up on these sites, and I tested them for metal uptake and found that there's some really common plants that were taking up high concentrations of the metals of concern, which is awesome. Those are great candidate plants to use in phyto extraction, where you plant these plants and they suck up the metals of concern so that you can remove them, cleaning the site. I also sequenced the fungi, and oops, and I probably don't have time. This might, basically what I want you to take from this is just showing the phylum, the dominant fungal phyla that were found on these contaminated sites. And 73% of them were from the Ascomycota, and they were just, you know, Ascomycota, again, they're not as charismatic. They're molds and yeasts and stuff, but they're really good at dealing with stress. And they, there were a lot of melanin containing fungi as well. Melanin is like this fungal armor that protects fungi from stress. So I thought this was really cool to just see. A lot of them were also petroleum degrading fungi. So it's kind of like they show up. They show up on these sites, they volunteer, and the reason they're doing that is because they can tolerate those conditions, those harsh conditions, and they can get the food they need from it. So I probably don't have time really to talk about too much about my current research, but I am focused on mycorrhizal fungi, so the plant friend fungi, plant symbionts. And some of the questions I'm looking at is how can mycorrhizae be applied to protect crops, like food crops, from taking up hazardous metals. So that's one of the things I'm looking at. I'm gonna have to skip some slides because just suffice it to say, as we've talked about, metals are a problem. They're in soils, including agricultural soils, and our food is one of the ways we get exposed to them. Yeah, I'm just gonna kind of try to move through this pretty quick, but so I'm working on a greenhouse study with some of these, some common food crops and some of these top phyto extractor plants I found growing around here. And I'm basically using indigenous mycorrhizal fungi, comparing them with an inoculant, like a commercial mycorrhizal inoculum, and then sterilized soil, so there's no fungi. And I'm also looking at, I mean, I think you're probably dealing with the opposite over in Georgia, probably more flooding than drought, but over here we're dealing with drought. And so I am looking at how these two different, how drought affects this. And okay, the part I really wanted to get to, so that greenhouse study is a lot more controlled. And you kind of need to do that, or it's beneficial to do that, more controlled study. I am using real contaminated soils from my partner sites, but I'll have a lot more control to see how these interactions work. The goal is, of course, to really be able to say, hey, can we have farmers, either with the indigenous AMFs that are already in their soils, mycorrhizal fungi, or with a commercial inoculum, block or stop these hazardous metals from getting up into our food. And under what conditions does that work? And are there the conditions in which that doesn't work? The second aspect to my overall research is I'm so excited about is field testing soil mycophytoremediation on large, heavily contaminated sites across Southern California. I have amazing partners, the city of LA, and lots of community groups and the regulators, the Department of Toxic Substance Control. So I'm really excited. You might notice I'm always reaching out to build those relationships with, government, community organizations, just to build relationships so that fungi are really incorporated into food systems, medicine, remediation, and so trying to remove those barriers and just create partnerships that establish fungi as this long neglected but very promising and very important aspect in these different fields. So I have four contaminated sites. 2 are future community farms and 2 are future parks. One of them is shown here. It's 45 acres big. It's a huge site. It was a rail yard. It has 25,000 parts per million of lead and diesel, super contaminated. And so we're basically going to test native metal hyperaccumulator plants I found growing on sites in the region that were taking up lead and other metals, comparing them against known phytoaccumulator plants that are not native, with indigenous and introduced mycorrhizal fungi and decomposer fungi, both native and introduced, comparing those with and without water. And that's paired with the greenhouse study as well. And so, yeah, a bit of background just quickly is that arbuscular mycorrhizal fungi, the generalist mycorrhizae, they help plants grow, and they have shown in a bunch of studies to be able to enhance the success of phytoremediation, or using plants to deal with contaminated soils, usually metals. Other fungi, this is oysters from the, I think this is from the study that Paul Stamets did in Washington, the transport yard with oyster mushroom. So it's several times oyster mushrooms have shown to be able to degrade diesel and other petroleum hydrocarbon contaminants. So how does phytomycoremediation work? Well, it just combines multiple fungi and multiple plants. So the plants take the metals up from the soil into their above ground parts where they can be removed. Helper mycorrhizal fungi around the roots of the plants help these plants grow and take up more metals. Beneficial decomposer fungi eat and break down diesel into non-toxic forms. And so together, we're hoping that this method will provide clean sites on 4 sites and be something that could be applied a lot more broadly. So I've partnered with smallhold mushrooms, a big commercial mushroom grower is providing the spent spawn. Right now it's a waste for them. We are testing again commercial inoculum for the mycorrhizae versus the indigenous fungi already there, testing these native plants. Whew. Okay, and then I just wanted to end on this last slide to go back to the Healing City Soils project. So I've been continuing to work on a community mycophyto remediation project back in Canada so again, the Healing City Soils, we identified that, yeah, lots of sites all around the city have metal contamination. And we really wanted to be able to offer solutions, like what can people do about it? What can the average gardener or farmer do about it? And so we're testing also native plants with mycorrhizal fungi and compost. And the project, this is one, we have several sites we're testing this on. We're including a backyard garden, a community garden, and a First Nations traditional food harvesting site. And the project is called The Ground Beneath Our Feet, Reciprocity and Soil Restoration. So we do have, I'll share the fact sheet, the basic bioremediation fact sheet I created through this project. But once we have results from this sort of demonstration remediation, for this more community kind of backyard grower type project, I'd be happy to share those, as well as findings from my larger studies, which are meant to address large, highly contaminated sites. And so with that, I really thank you for inviting me to share some of what I've been working on and learning with you. I have my contact info here. Please don't hesitate to reach out if you want to connect in some way. And so, yeah, I'm just gonna check that there were, thank you, Sam. Jargon. Yeah, AMF, mycorrhizal fungi.
Sam wants to point to me because I'm doing most of the typing in the questions actually.
Perfect.
But you know.
Okay, awesome.
So everybody take a good screenshot or a picture or something so you have her info.